Hi everyone, my name's Christina, and first off, I just wanted to start off by saying that I'm so sorry I can't be here to present to you live, but I do hope you enjoy my presentation. For my subject expert project, I focused on mesenchymal stem cells, or MSCs, therapy to specifically target the regeneration and restoration of ovarian function. For an overview of my presentation, first I'll provide background information regarding anatomy, causes of female infertility and associated treatment plans, as well as address the gaps and limitations to current treatments. Second, I'll review research that has been performed to this day regarding MSC stem cell therapy. And lastly, I will conclude with suggestion for direction for future research. The major female reproductive organs are the ovaries, fallopian tubes, uterus, cervix, and the vagina. The ovaries can be seen as the tan sac-like structures on the left and right of the image above. A healthy female will have two ovaries as shown, and the ovaries are the site of follicle or egg development. The fallopian tubes, as shown, are passageways from the ovaries to the uterine cavity. The uterus can be seen in the middle of the image on the right-hand side of this slide. The uterus, as we know, is the location at which the fertilization and gestation occurs between an egg and a sperm. From an anatomical perspective, the uterus can be viewed as two parts: the corpus, which has the endometrium and the myometrium layers, and the cervix. The cervix is located at the bottom of the uterus and is essentially connecting tissue between the uterine cavity and the vagina. Lastly, the vagina is shown at the bottom of this picture and is fibromuscular structure that extends from the cervix to the vulva. Since my review is specifically on the ovaries, I wanted to take some time to walk through the anatomy of the ovaries, go into a little more depth about their role, and also review how they function as organs. There are three main layers in the ovary: the endometrium, the myometrium, and the serosa. The ovaries contain germ cells, also known as eggs, as well as somatic cells, which include granulosa and stromal cells. In the body, the ovaries are responsible for hormonally releasing eggs monthly in postpubertal females, as well as regulating hormones in the body. The function and relationship with the endocrine system. The main events in the ovaries are controlled by two hormones, FSH, or follicle-stimulating hormone, as well as LH, luteinizing hormone. The hormone levels in the ovaries are controlled by the pituitary glands and the hypothalamus. Now that we've covered female reproductive organ anatomy, I want to outline the clinical need for research on female infertility. According to the CDC, approximately 10.9% of women experience impaired fertility. The CDC defines infertility as the inability for a woman under the age of 35 to conceive after 1 year or longer of unprotected sex, or the inability for a woman over the age of 35 to conceive after 6 months or longer of unprotected sex. The causes of impaired fertility can be broken down into three overarching categories: endocrine system, anatomical abnormalities, and iatrogenic infertility. As we know, the endocrine system is responsible for regulating and releasing hormones in our body. Hormonal imbalances in females can lead to fertility difficulties. The most common treatment for females suffering with endocrine-related infertility is hormonal therapy. Anatomical abnormalities encompass both congenital or sustained injuries. The treatment can either be surgical or hormonal depending on the location, the type, and the severity of the abnormality that's leading to infertility. Iatrogenic infertility is caused by sustained injuries from medical examination or treatment that have been performed by a healthcare provider. Iatrogenic injuries can be caused due to both physician negligence and or as an unavoidable or unpredictable outcome caused from a medically necessary procedure. While the treatment protocols vary depending on the type of injury, for the scope of my presentation, we will be focusing on chemotherapy-induced infertility, which is considered an unpredictable outcome or unavoidable outcome from a medically necessary procedure. I do want to note that although the causes of infertility can be easily broken up into these three categories, infertility is generally way more complicated, complex, and integrated. Therefore, it is likely that one's fertility is a combination of the three types of infertility listed below or above. As most of us know, chemotherapy, sometimes used in combination with radiation, is the most common form of cancer treatment performed today. Chemotherapy can be designed to damage healthy cells as they kill cancer cells. This is known as traditional chemotherapy. Or it can be targeted, where chemo is used only to kill cancer cells and block them from dividing and therefore spreading. When it comes to chemotherapy's impact on fertility, a ranking system has been created to classify chemotherapy drugs according to their impact on fertility. The highest risk chemotherapy drugs all contain alkaline agents. The ones that will be mentioned in this presentation are cyclophosphamide and busulfan. Now we'll review the impact chemotherapy has on the ovaries. While the exact mechanism at which cyclophosphamide and busulfan causes ovarian toxicity, research has been done to understand the specific effects they have on the reproductive organs. There are two types of ovarian failure that can occur: primary and secondary. Primary ovarian failure is when the follicle reserves are depleted within the ovaries. Therefore, the dormant eggs that reside in the ovaries and await maturation are adversely impacted. Secondary ovarian failure is when there is a large reduction in stroma and granulosa cells inside the ovaries. Agents in chemotherapy cause DNA damage via cross-linking within these cells. Therefore, protective mechanisms in the body initiate cell death to those cells via apoptosis. Because granulosa and stroma cells are very important when it comes to regulating hormones, the depletion of these cells causes inadequate regulatory signaling between the reproductive organs in the pituitary gland and hypothalamus. Females who are diagnosed with cancer and face chemotherapy treatment are often provided with options when it comes to preserving one's fertility. The current fertility preservation methods either take proactive or preventative measures. Proactive methods of ovarian preservation aim to save a female's eggs, either fertilized or unfertilized, or ovarian tissue, with the hope for implantation after chemotherapy. Cryopreservation of eggs is performed to save the eggs in the ovaries. Preservation of ovarian tissue is performed to save the healthy tissue in the ovaries, with the hope to reimplant into the patient to encourage healthy growth of cells. Preventative measures of ovarian preservation aim to prevent the damage of the ovaries or to the ovaries during treatment. Two examples are ovarian shielding and ovarian suppression. While the medical field has many options to preserving a patient's eggs or ovarian tissue, there are no clinical options today when it comes to restoring a patient's ovarian function. Next, I'll review the ways in which mesenchymal stem cells, referred to as MSCs, have been studied to be used as an option for ovarian restoration in patients who have undergone chemotherapy. There are two important questions that must be answered before we move forward. 1. What are mesenchymal stem cells? And 2. Where do they come from? MSCs are multipotent adult stem cells, meaning they have the capability to divide into one or more different types of cells in the body. They originate from stromal cells, which are cells that become connective tissue of any organ. They must also fit the following criteria. 1. They must be plastic adherent. 2. They must express clusters of differentiation, abbreviated as CD, at CD105, CD73, and CD90, while lacking expression of CD45, CD34, CD14, or CD11B, CD79A, or CD19, and human leukocyte antigen, HLA, class II. And thirdly, they must have the ability to differentiate into osteoblasts, adipocytes, and chondroblasts. MSCs are traditionally found in bone marrow, but can also be isolated from cord blood, peripheral blood, fallopian tubes, and/or the fetal liver and lung. MSCs have a wide range of applications and potential. Cell therapy of MSCs have been hypothesized to have a positive impact in restoring ovarian function in women undergoing chemotherapy. In clinical studies that have already occurred, specifically in rats and mice, we have seen positive results in restoring ovarian function. I will now review an important study that was done and share the discoveries that were made. In this study referenced, the test species were mice. There were 3 test groups in this study. Group 1 was the control group. Group 2, the mice were treated with chemotherapy as well as a placebo stem cell treatment. And Group 3, where the mice were treated with chemotherapy and the correct stem cell treatment. The study lasted approximately 8 weeks from the first site of injection of chemotherapy. They were testing markers at week 2, 4, 6, and 8. The chemotherapy drug that was used was cyclophosphamide as well as busulfane, which are both alkaline agent-containing chemotherapy drugs. The bone marrow transplanted into Group 3 of this study was collected from a human female. The metrics that were measured were subject weight, vaginal smears, blood tests, hormonal assays, and 1 subject was sacrificed each time marker and was examined. The large takeaways from the clinical study referenced are as follows. MSCs showed a protective effect on apoptosis in stromal and granulosa cells. 2. In a previous study conducted by the same researchers, it was demonstrated that MSCs can reactivate dormant folliculogenesis in rodents that have ovarian dysfunction. 3. Estrogen levels taken from vaginal smears showed changes in the test group when the control group remained the same. 4. Reproductive organs and estrogen-responsive organs increased in weight. 5. An increase in follicle count was observed at each time marker. 6. Increased E2 and decreased FSH were observed. And finally, there was an observed rescue of infertility phenotype in treated mice group compared to the untreated mice group. Lastly, I'll spend some time recapping the key takeaways from this presentation so far and briefly touch upon future research. I wanted to recap on highlights of my presentation so far. First, the scope of the research in this presentation focused on females undergoing chemotherapy who might want options when it comes to their infertility after treatment, specifically in restoring the function of their ovaries. This research is very important because females are 38% less likely to become pregnant after cancer and diagnoses. It has also been shown that a loss of fertility can lead to a decline in mental and emotional health. And lastly, I wanted to reiterate that we, as a scientific community, have many ways to preserve eggs for possible fertility in the future. Yet it is necessary to research how we can regain ovarian function after that treatment has occurred. Looking forward, 1, we need to understand how MSC cell therapy acts on human beings. One assumption that was made in the clinical study that was referenced was that if human stem cells were to function xenogenically in mice, then it was expected they would function as well or better when administered to humans due to their immune privilege status. Yet this assumption needs to be verified via human clinical trials. Secondly, it would be important to further study how MSCs impact the long-term rate of ovarian health. Third, we need to understand the effect of MSCs on prepubescent females, therefore females who might undergo chemotherapy prior to the age of puberty. Lastly, it's important to understand how the duration of chemotherapy impacts regenerative rates driven by MSC therapy. Thank you for listening. Any questions?